For this nice introduction. Of course, I also would like to thank the organizers, Danielle, Morten, and Alex, for what I think is, of course, again, a wonderful meeting. And it's not only the science, but it's really also the interpersonal context. I mean, in the age of Zoom, we sometimes forget that we really need to meet each other live. I think that's really important for science. So let's talk somatic mutations. I can't talk about anything else anyway, I guess. So now this is very old theory. Of course, it remains important apart from interventions and translation. It remains important to think about causes of aging, I guess. Let me see if I can get a pointer. And I can. So this was proposed already in the 1950s, actually shortly after Watson and Crick identified the structure of DNA as the heritable material. Of course, mutation, the concept of mutations was known since about 1900, but then as altered genetic traits. And nobody really knew the basis of it. And because of Watson and Crick, we started to realize that mutations were changes in DNA. And then very shortly thereafter, first Gino Faya and then Szilard came up with what I think is really an elegant theory. They realized there was always DNA mutations because there's always some background radiation. Now, of course, we know because of Thomas Lindahl's work that there's a host of sources of spontaneous DNA damage which are then turned into mutations. But in those days, they couldn't really know that. But they formulated this hypothesis in terms of Gino Faya, mostly because he was well aware that the risk of cancer increased exponentially with age, and he suspected that cancer was due to mutations. You should realize that he couldn't really know this. This was only known in the 1970s. It was definitely proven that somatic mutations are the cause of cancer, and cancer increases with age. Now, just to make very clear, mutations is not DNA damage. Many people continue to think that it's the same. DNA damage is just chemical or physical changes in the DNA structure. It can be chemical adducts, it can be breaks, single-strand breaks, double-strand breaks, loss of bases. And DNA damage is extremely frequent. You can think of probably somewhere in the range of 100,000 lesions per day per cell. That's an enormous amount. So that tells you something, namely that DNA is not a stable molecule under physiological conditions. It's only stable because of the enormous network of DNA repair processes, which is extremely conserved. It's probably one of the first genetic traits that was actively selected for in life. Probably we're talking about the early replicators. And in a way, it didn't change much since those. It's a very conservative system, very essential. And it probably a very large part of the coding capacity of our genome is only coding for genome maintenance systems in one way or the other. So I actually decided to talk about something else that may be evident for my abstract. I was going to talk about the influence of negative selection processes in the pattern of somatic mutations in relation to aging. But I thought that was basically not so suitable for this audience. So instead, I want to discuss with you a little bit about predictions that you can make of this somatic mutation theory of aging, if it's really causally contributing to the aging process, other than cancer, of course. So first, of course, you want to see that mutations really accumulate with age in human and animal tissues, not? Second, everybody is talking always, and I get these questions, that's why I put them on the list here. Does caloric restriction reduces mutation load? That's what people always ask me. Second, can you see that very long-lived species have cells that have a very low mutation rate, and rapidly aging species, the other way around, you see a high mutation rate. Then, of course, as Daniela pointed out just a moment ago, many of these human proteroid syndromes are actually caused by genes involved in genome maintenance, defects in genes involved in genome maintenance. So you want to see that the mutation burden really is higher in cells from these proteroid syndromes. And finally, that's maybe a little bit more complicated, you want to see that mutation rate in germ cells is very low, because germ cells in contrast to your somatic cells, have to not go one lifetime, but forever, not? So in principle, mutation rate should be 0 in germ cells. In stem cells, it should also be low, because they have to give rise to new differentiated cells. Now, in order to do this, you need to have a method to actually analyze, quantitatively analyze somatic mutations. And those methods were lacking until about 10 years ago. You would say, well, what's so difficult about it? You just take DNA from a liver or a brain and you sequence the hell out of it, and then you can find your mutations. Unfortunately, that's not the case, because if you actually do that, and this is your bulk tissue, and here are your reads, this is of course already taking into consideration, we have advanced sequencing methods, you have all these different reads, and we know now, and this is because I tell you, that here there's a mutation in one cell, which is not present in the other cells, because all these cells have different mutations. All have mutations, but they're all different. Now, you are very lucky, because you should screen through maybe, I don't know, maybe 1,000 reads, but you find it already after a fairly small number of reads. This is it, but you only know because I tell you. Not? But look at these. These are sequencing errors. Sequencing error rate is about 1%. So how would you know if this is a mutation? This is why you cannot see somatic mutations by just sequencing tissue. So you can take a single cell approach, as we've done, and when you do that and you do a whole genome amplification, you can see the mutation in half of the reads. Only one of the 2 alleles is affected. This actually is a SNP. SNPs, of course, should be present in all the cells of a tissue. That's why we see it here, heterozygous SNP. There are artifacts, but we modified these two, this talented couple in the lab, modified this assay to make sure that we weren't bothered by artifacts. Now, since single cell sequencing is expensive, you have to do a whole genome sequencing of multiple single cells if you want to know the mutation rate in a particular tissue. So Alex Maslov in the lab, he actually developed a single molecule approach based on a very nice principle that Larry Loeb, University of Washington, came up with originally. He realized that artifacts showed a mutation only on one strand, while a true mutation should have the mutation opposite on the 2 different strands. And you can distinguish between that by sequencing single molecule. And I mean, Alex modified that to make the assay actually practically applicable. And it works very well. You can see that here. When you do dose response with a powerful mutagen, you see that there's an increase in mutation frequency. We compared it to the single cell approach, which is like the gold standard sort of nowadays. And it all worked out well. He modified it later again, this has not been published yet, to actually also detect genome structural variations. Using single cell analysis, we cannot see those mutations. And those are highly impactful. Those are like large deletions or translocations. Now, the first sort of prediction is that, yes, mutations should accumulate with age, and they do. This was actually not so well known, because before, let's say 10, 15 years ago, we could only rely on germline mutation frequencies, which are very low. I will show that to you later. So when we first published an S, that was still with a mouse model, and we came up with these high mutation frequencies. I had great difficulty in getting it published, because he said, well, you are 10 to 100-fold too high, and they're all artifacts. But eventually this worked out. Now, in reality, somatic mutations are much higher than germline mutations. Now, you can see that they vary. These are 3 different organs in B lymphocytes, the blue ones. Each dot is a single cell here. This goes from a couple of hundred per cell in very young newborns, so to say, to a couple of thousand in centenarians. Now, in liver, this is far higher. You can reach like 8,000 mutations-based substitutions per cell. So that's quite a lot. And the lung is a little bit in between. If you look in lung cells from people, and we have done that, who smokes, then there are more somatic mutations. So again, just for your information, don't smoke. It's not good, you get cancer out of it, okay? Now, these are small insertions or deletions. So that's about 10-fold lower than base substitutions. Why? Probably because they are more impactful. When they hit a gene, they knock it out, so to say. So maybe that is the reason why they are lower. Same for germline mutations, you find that same thing. Now, the first question sort of was already resolved, actually, with our original mouse models, where we use selectable markers. Yes, caloric restriction does reduce your mutation burden. That was shown by multiple papers. this is our own paper here, where we also looked at dietary restriction in combination with dwarfism. So both dwarfism, which is like a growth defect, also increases lifespan. You see that the mutation frequency is lower, and also in caloric restriction it's lower. And when you combine it, it's even lower. Now, there are other papers about that now, so that's sort of clear. Why is that? Well, probably because caloric restriction reduces the metabolic source of DNA damage. That's at least what I think. We don't really know this for sure. Now, the third question would be if the somatic mutation rate really correlates inversely with species-specific lifespan. And together with Vera Gorbunova, she has a beautiful arsenal of different rodent species, different lifespan, from mouse to naked mole rat, and she gave us primary fibroblasts from these species. And we demonstrated indeed that when you have a cell from a naked mole rat, then the mutation rate is significantly lower than that in the mouse, for example. And also, when you treat these cells with a low dose of a mutagen, you see less mutations in cells, or actually no mutations at all even, in cells from a naked mole rat as compared to the mouse, where you see a lot of mutations. A little bit later, this was confirmed by another study with a whole different set of animals using also a different method. And that they found that they're essentially the same result. So yes, there's an inverse correlation also. When you live longer as a species, you have lower mutation rate. Now then, this comes back to what Daniela just said in the talk. Progeroid syndromes, like Werner syndrome and many others, they are mostly, maybe even exclusively, based on genetic defects in genome maintenance genes. For example, Werner syndrome is a RECQ helicase defect. So the question then, if this is accelerated aging, do you see more mutations? Now, in case of Werner, you probably suspect not so much base substitutions, but genome structural variation because of the type of defect. And Elizabeth Pond in my lab, a student, actually obtained blood samples from Junko Oshima, this University of Washington. They have a beautiful collection of Werner, not so much cells as blood, so we could only extract DNA. So here we used our single molecule assay. We had no single cells. And Ellie compared blood from young people, old people, and Werner people. These are the Werner homozygotes. These are the Werner heterozygotes. And we were a little bit surprised about a dramatic increase in genome structural variation. I'm talking about large deletions, translocations, and so on. And also because there's no difference between the heterozygous and the homozygous. The heterozygous Werner do not show, as far as I know, any clinical symptoms. So this is strange. However, these are much older than these. Many of them are their parents, actually. So you can imagine that when they were still fairly young, the increase was sort of low. But it also implies, therefore, that heterozygous are at risk in a way. Not, as far as I know, nobody has ever done a large study. In Japan, there are many Werner syndrome patients, and there should be a great number, I don't know, maybe 1 in 100 or 1 in 200 people who run around with a heterozygous mutation for Werner. On the other hand, Japan also has the longest lived individual. So, but still, it's very clear results. Those are mostly translocations. You can see that here. Significant increase in translocations. So then sort of, well, a little different. Now, would you expect that mutation rate in germ cells is very low? As I said, it must be, not? Otherwise, we would not be here, I guess. Our germline must go on forever. It cannot be that high. You have, of course, selection. That's a factor 2, but it cannot handle it on its own. Must have a better genome maintenance. That was also predicted by the disposable soma theory in this paper of Tom Kirkwood in Cell. He basically argued that the immortality of the germline just requires a high level of genome maintenance. And when you compare it to somatic cells. Now, so that was an interesting thing to test, because now we could test it. Now, a student in the lab, Brandon Milholland, actually first compared germline mutation rate with somatic. He could do that now after we published the single cell assay. And he could then basically take fibroblasts from mice and from humans, and then measure the somatic mutation frequency. And I already pointed that out to you. We did that also in different organs. Here we plotted the mutation frequency per mitosis. He could calculate that because we know, of course, the number of cell divisions per generation, or at least could estimate it. And what you can easily see is that the germline mutation rate is indeed 10 to 100-fold lower than the somatic mutation frequency. And in mice, you see a somewhat higher mutation frequency generally than in humans, about twofold in fact. Here, this experiment, the difference is not that large, but it's generally almost twofold. Now, you can of course confirm that, not? You can just go directly to germ cells. We didn't do sperm, but we did do oocytes. This is Joseph Cherniak, who's a clinician, he worked in the lab for a while. He's from OBGYN, and he basically collected oocytes and then did the single cell analysis to see how many mutations there were. And you can see here, when he took those from women in their early 30s, of course they got these oocytes from women who came for IVC, no? That's how they get these women and were able to get the oocytes. For IVF, I mean, in vitro fertilization. So you can see very clearly that the mutation frequency is indeed very, very low. It's about 200 mutations per cell. It's significantly lower than what we see in fibroblasts. You see a dotted line here. We always include a fibroblast, somatic cell there. It's clearly much higher. Now, somewhat more surprising in a way was that women who were only like less than 2 decades older, you see a very high mutation frequency. It's actually higher than most somatic cells. Now, that was a little surprising to me, but not to the people from OBGYN. They said, well, it's well known because aneuploidy we can measure. Aneuploidy rates also going up dramatically in women when they grow older. This is just indels. It's just the same story, it's just lower. And this is basically tells you that the intrinsic fertility of oocytes goes down dramatically. This is the baby take-home ratio, as they call it, and it goes really down dramatically with age here. So it's very difficult to actually get a baby when you are pretty old. Of course, it's also selection that plays a major role here, so it can be done. Now, a related question here is, of course, the stem cells, not? Do we see that in stem cells as well? And Johanna Heid in the lab, a postdoc in the lab, decided to make induced pluripotent stem cells from fibroblasts, from just primary fibroblasts, and then measure the mutation rate. This is point mutations again, also using single molecular analysis. Now, look in blue. There are 6 clones, IPS clones that she obtained from the mother population. This is measured 3 times here. And you can clearly see that the mutation frequency is very low in the IPS clones, much lower than in the original population. Now, you can guess why that is, not? Cells with a high mutation burden are definitely not reprogrammed. You need to have a cell with a low mutation load in order to get reprogramming. That's what happened here. She also treated each of these cells with a low dose of ethyl nitrosourea, powerful point mutagen, I mentioned it before, and then she calculated what was actually the relative gain of mutations. In other words, the question was, the IPS cells, once they are IPS cells, did they get a better genome maintenance and to suppress mutagenesis? That seems to be the case. You can see that in the original source population, the same dose of ENU induces significantly more mutations than in the iPSC. So it sort of opens up the exciting possibility that when you make stem cells, you sort of change something in your genome maintenance, I guess. So the conclusions are that, yes, somatic mutations do accumulate with age, and they do so in a tissue-specific manner. The question is, of course, what exactly are they doing there? I will come back to that in a minute. Somatic mutation rate in mice is reduced as expected by dietary restriction. It's inversely correlated with species-specific lifespan. It's higher in blood, at least. We haven't done any other organs from Werner syndrome patients, and it's significantly lower in induced pluripotent stem cells than in the source population. And mutation rate is indeed very low in germ cells. Now, what are we doing now? Which direction are we going to take? Well, of course, we want to know how, if they have a functional effect, how can that work? And quite a long time ago, we published a paper where we found increased what we call transcriptional noise in cells. We compared young and old mice, different cardiomyocytes, and of course, when you have young mice, every cardiomyocyte should express a particular gene transcript to exactly the same level. Not if they start to fluctuate, it's not good. Well, they do start to fluctuate because in old mice, we see that the same gene is expressed to different levels between different cardiomyocytes. So there's an increased level of transcriptional nodes, by now well confirmed by many other studies. So what we are planning to do now, or are involved in doing now, is to do single-cell multiomics. Each cell we sequence genome and transcriptome to look at consequences of particular mutations. And then we want to know what is the potential functional impact on gene regulatory networks. And we do that with Laszlo Barabazi, he is here. And you should go to poster 58 to see some already interesting results by Joey Ehlert. Now, second, and this is work by Bjorn Schumacher. You wouldn't know, because of course he didn't talk about that. He didn't tell me he wouldn't do that, but at any rate. So he discovered that this dream complex, and I honestly wouldn't be able to tell you what DREAM means here, but it is involved in cell cycle regulation, and it's active in somatic cells, and it seems to suppress DNA repair activities. So what we want to do now is to see if this potential master regulator of repair is actually capable of reducing somatic mutation burden in cells, because then we would have a way to keep mutations low, sort of to adopt somatic cells to what we now observe in stem cells and in germ cells. So these are all the people who did the work. Moonstok, of course, my lab technician, taking care of us. Alex Maslov, I already mentioned that he invented the single-cell analysis. Xishiang Sun is really our computational person. Zenchi did the lung work. Ellie, I already told you about it. That is the Werner work. And Johanna, and Joseph, I already introduced them. Now, my collaborators, there's of course my dear friend Judy Campisi, who recently died, and that was an enormous loss for the whole scientific community, but certainly for one of her best friends, and that's me, because I used to talk to her quite a lot about science and about life and everything, and now I can only imagine to talk to her. Thank you.
Thank you, Dr. Vijg, for this amazing presentation. We have time for one or two questions.
So Jan, I wonder, in your you suggested that the high mutation loads would impair reprogramming, but have you considered testing whether that these high mutation rates could be correlated also with high damage levels, for which transcription stress, for example, could be a parameter? So in other words, if you look at single-cell transcription stress and mutation rates, do you see the same effect between the reprogrammed cells and the...
Yeah, well, I'm sure you know that it's very difficult to measure spontaneous DNA damage. We can, of course, measure induced DNA damage. We could induce, with very low levels of ENU, damage in cells, but then how would you then distinguish between the effect of mutations and damage? So we have not done that. And I think DNA damage is really a difficult thing to measure. I wouldn't know.
But transcription stress could be an indicator, right?
Well, true, but do we really know that transcription stress comes from DNA damage? So, right.
Last question. Yeah.
It's on. Yes, it is now. Great talk. Thanks very much. I'm sure you would find correlations between somatic mutation rate and other hallmarks of aging, but do you have either hypotheses or experiments to see if somatic mutation actually could cause the other hallmarks of aging?
Yeah, I mean, what you're asking essentially is, can we directly look at particular somatic mutations in cells and then see if they could have such an effect? Well, we haven't really done that because it's not that easy. So what we are in the process of doing right now is to look at human T cells. Their T cell activation is known to go down with age. And we are now doing this single-cell multi-omics experiment where we try to link particular mutations in gene regulatory sequences to the network of genes that controls T-cell activation. But we haven't done that. This is extraordinarily difficult. We are doing this already for quite a while, and thus far we made some progress, I think, but it's always difficult to know if this is actually going to work.
So we are now closing this session. So
Good.
thank you all for your contribution, and thanks again to our speakers.